Roger, where do you come from?
I just hit the record. It's
Yeah.
blinking. It's good to go.
Okay, cool.
See.
And then this is how I
Which
zoom
thing
in
is
and
that?
out. Okay. If I need to.
Yes. Yeah, and you can turn it.
Turn this down. Yeah. Pretty short.
Yeah.
Thank you.
Vertically
Okay.
damaged.
All right.
All right.
And otherwise.
And other, yeah.
Yeah.
And otherwise.
Okay, thank you.
Uh-huh. All right.
Thanks.
Just give me a call. That means you go up there.
All right. Anytime.
All right, so let's begin. So today, we're going to be covering how to do RNA-seq with iPlant, specifically internal to one of its feature flagship products known as the Discovery Environment. For people that were here earlier in this workshop series, we have used the Discovery Environment for data management. The other half of that platform lets you integrate pretty much any tool that runs on the command line of Linux, and then use the data that you have in your data store to run through those specific tools. When tools are integrated within the Discovery Environment, people have the opportunity to design a user interface that's presented within that environment. And what Roger has done, in conjunction with some other people, are integrate a series of tools that let you perform RNA-seq analyses. And so this is now bringing together the entire other part of what the Discovery Environment can do. Now when these applications run, they run on a local compute cluster here, so we can get a lot of scalability. So if you tend to have maybe 50 or 100 experiments that you do need to analyze, we do have the capabilities behind the scenes to scale accordingly. Some of these can, of course, scale even larger on high-performance computing, and Roger may or may not cover that today. The other part of it, which will be at the very end, is doing similar kinds of RNA-seq experiments or analyses inside of a platform known as Koji. Koji is a comparative genomics system that my group maintains, and what we do in order to get computational scalability is we're using a lot of the underlying infrastructure components of iPlant. This means that when you log into Koji with your iPlant user credentials, Koji now can do operations on your behalf, including getting data out of your, and only your data store, and then using iPlant's infrastructure to get a lot of the scalability it needs behind the scenes. And so with that, I'm very happy to I'm very happy to introduce.
My producer.
Very happy to introduce Roger Barthelson. He is one of iPlant's scientific analysts, which is a team of super PhD level scientists that work with iPlant in order to help facilitate the community of researchers to best utilize its underlying technologies. Roger has specialized in a variety of genome assembly annotations as well as functional genomics, as you'll learn today. He comes from the very first state of the United States, known as Delaware. I, unfortunately, came from the California public school system, and so that was one of my puzzle pieces as a young kid that had a whole bunch of other states attached to it. And I really don't know much more about Delaware.
It was probably the one you lost, because it's tiny.
Well, but they joined it together with others, so it was like, here's the
Yeah,
Northeast.
that was humiliating. Stuck with New Jersey and Maryland.
Yeah. So I do really want to thank Roger for making it in today. As you can tell, he is differently able for a little while, and he has this awesome high-performance laser-guided mobility system. Don't upset him. This thing, I think, is tied into infrastructure, and it can zoom around and make sure it will make sure you keep doing your work if you're not doing your work. And so with that, Roger, I hand it over to you.
Thanks, Eric. I hope that my relatively non-dynamic presentation today, since I will be sitting through most of it, will be stimulating enough or of value enough to you that you'll pay some attention to it. As Eric said, we'll be talking about RNA-seq, and I'm going to call on some of you to help me teach this class because I only have one foot that's functional right now. So, and sometimes I like to think on my feet. So, with that, what is RNA-seq? Um, let's see. Up here, since you guys sat in the front row, that means that you know all the answers, right? Excuse me if I point. You, what is RNA-seq?
What I understand, suppose the RNA has the all gene information over there. So you are actually going to sequence bulk of gene information from RNA to cDNA and then.
Right. That is more or less correct. We use it for gene expression studies by sequencing the reverse transcribed DNA or the reverse transcribed RNA, which becomes cDNA. I don't think RNA sequencing is commonly used at all, but I understand that it is possible. So just so that you guys understand, when we do talk about gene expression, the general concept is that we are talking about what, trying to look at what programs are being turned on within the tissues that we're studying. So we are looking at samples that typically reflect a certain tissue type, a certain cell type, or whatever the hell you guys stuck in the test tube that day when you prepared your RNA. So you have to keep that in mind. There are different programs for each individual cell, so you're looking at a combination of different signals. And each cell has a lot of different signals going on, RNA being produced. A lot of you understand that, but it's important to sort of keep that in mind as you go through the process. What are we looking at? Why are we doing this? And the other aspect of that is, too, you don't want to forget that when you're looking at the RNA, we're looking at what is transcribed, but we are not looking at this, often when we interpret these things as being what's translated, because what's translated is not always exactly what is being transcribed. And if you focus, as I have at times, on looking, for example, at what is going on in the nucleus, you'll find that there are a lot of nuclear forms of RNA that don't make it out of the nucleus. Some of them may be actually transcribed there for certain purposes, maybe even for producing proteins. But some of them are only there for editing purposes. That is, they're the forms that eventually get edited down to what goes out into the cell for actual production of the proteins. So, and beyond our messenger RNA, which is most of what we're looking at, then it comes down to it's not just the messenger RNA, but it's also what controls the message in the form of interfering RNAs, microRNAs, silencing RNAs. So, there are a lot of different types of RNAs, and of course ribosomal RNA, which could swamp your signal. So a lot of times you start out by getting rid of the ribosomal RNA. So, but you have to keep in mind that sometimes these things are present, a lot of the times they are not. But when you look and examine ultimately the results of this, which is the idea, we're not just doing this to go through the process of RNA-seq and get an answer. We have to have a strong idea to begin with, where we start and where that leads to in order to get whatever answer we want to get out of the data. We have to know exactly how each, keep in mind, at least in the back of our minds, how each step is affecting what we actually see in the end. For example, when you prepare your RNA, most of the time, you're going to throw out all the microRNAs. It's just not going to be captured by the processes you typically use to capture messenger RNA. So just keep that in mind. Usually you have to specifically start with a preparative method that does give you that microRNA. So let's get into some of the nuts and bolts, because a lot of iPlant is nuts and bolts. Let's face it, it's an infrastructure. It's a way to help you analyze your data. And the first question I put up is, what if you have a question? What if a question comes up along the way? What can you do? You can always email me and ask a question. You can always ask Eric if you see him, or Jeremy over there, or Nicole even, if you can catch her. But what else? I mean, what is your first line of questioning if you have a question? Google. Smart person. A lot of people don't think of that, oddly enough. But obvious, you know, if you grew up with the internet, which I didn't, but you know, I learned pretty quickly. If you have a question, just put it in Google. You know, if you're working with a piece of software, just, you know, like we'll be using Top Hat, it's one of them. Just put it in the search box, Top Hat. If you want, you know, a very in-depth definition of how to use the application, and what are all the options, what do they mean? Top Hat, manual, maybe RNA-seq, just to tie it all together. But chances are it'll come up with your answer. So just keep that in mind. It's always easy to find an answer. And so again, when we're starting an RNA-seq project, you start with our organism, whatever it is, and a lot of the time you may be starting with a Arabidopsis up there in the far left-hand upper corner, or you may be studying something more exotic and new, and something that nobody else has sequenced before. And so your problems can be different, technically, what you're dealing with. You may be using different sequencing systems. Illumina, Ion Torrent, 454, PacBio, these are all ones that may be used. Most of you will probably be using Illumina system because it's cheap, high throughput, very available, pretty reliable. But some of the other systems have value too. And with time, we do have at iPlant support right now for Ion Torrent and 454. PacBio, you're more or less on your own, but we do have resources that you can use to help you. But as soon as we can, we're going to be providing PacBio read support also. So keep that in mind. So I think along the way, I haven't been here through all the workshops, that once your reads are ready and you know that the sequencing facility has produced them, and maybe you already have them on your laptop or your desktop, if you're smart and if you paid attention to what was described when somebody was talking about the data store, then maybe you already got your login for iPlant. I assume that you guys have already done that. That's pretty easy. And maybe you've put some of your data in the data store. But that's where your data will need to be in order to use iPlant resources. And I think also what has been described are ways of putting your data into the data store, and if you need to pull a file out in the end, to pull it down to your desktop or wherever. So the assumption is that you're working with data within the data store. So then it just comes down to, what do you do? I mean, how do you do RNA-seq at iPlant? And this again comes down to the different steps, but we've already talked about, well, what we're looking at as far as transcription, the sequencing process. Ultimately, we're going to be looking at fragmented reads, and those are going to go, and supposedly those are in the data store. They might be paired reads, and if some of you may understand that, and some of you may not, but it's an important concept to understand, even though it's just technical. Most of their sequencing systems provide, like iSeq 2000, pretty commonly used these days, a lot of data that comes out of that, an Illumina system. That offer the capability of paired reads, because you get your reads coming out as pieces. They're all samples randomly taken, hopefully or theoretically, of your RNA sample that was reverse transcribed. So fragmented, essentially, pieces of your cDNA. And so what that means is that ultimately you're going to have to put all those reads into some kind of intelligent array of information to make sense of it. You have to piece them back together, literally, because ultimately you're interested in the transcripts. Each read is a sample of a transcript, and mostly, or often, let's say, a sample of just one piece of a transcript, one exon within a transcript. So each read is really often just a sample of one exon. Occasionally, they'll bridge between two exons, and the applications, when they're good for use for RNA-seq, will often recognize, okay, this piece that I'm looking at bridges across, and so we're looking at not only information on two exons, we're looking at the actual junction. And junctional information is especially helpful because it tells you how to piece your reads together, provides a bridge, so to speak. So this is what we do when we do RNA-seq. We do, first, we analyze the quality of the reads and we clean them up. We're not going to be doing that today, unfortunately, because if you worked with me and you did what we are going to do today, which is just take your reads and start analyzing them without cleaning them up, I would get pissed off, because one of the things that I've learned, and I was by far not an expert when I started doing this stuff, is I learned the hard way that if you don't clean up your reads, you're putting a lot of crap into the analysis, and you're screwing up the process. Often on the ends of your reads you have adapter sequences, and you don't think so, but you often do. And you also have low quality reads, because all this information that comes into the reads is pulled off of plates that are fluorescent or light captured on film or some, or digital array. But the information is sometimes really weak. And if the signal is really low, then it can have misinformation. And you don't want that. Any misinformation ends up producing a lot of extra junk in your attempts to piece together your reads into transcripts. So one of the things key things to understand with the method that we're using today, the tuxedo method, tuxedo because it uses bow tie, top hat, cufflinks, and then the oddly named cuff dip. Although it describes what the fact that it's doing the final differential look at expression. But you begin usually with this method, or always with the tuxedo method, to map your reads to the genome. Why do you use the genome? Why do you even need the genome? You don't. Technically speaking, you don't. Because the only thing you really need to know about is RNA sequence. The genome sequence is more complex. It has all the introns and everything else in it. But, and that's one of the things that got me going when I started trying to analyze the data. Well, why can't I just map it to the transcripts? And you can. And sometimes you have to if you don't have a genome sequence at all to work with. But if you do have a genome, it's convenient to work with the genome because it ties your reads to where that transcript came from, the ultimate source, the DNA, the chromosomes. And often that's the most accurate measure of what the function is, or at least a way of getting at the function. What are the genes? Yes?
Is there a good way to visualize when you've mapped a transcript to a genome? Are there
Yes.
programs to actually visualize the chromosomes that are there and then to see where your reads are mapping down onto the chromosomes?
Yes, and Eric is going to show you that, I think.
Okay.
Unfortunately, the more dynamic and interesting part comes at the end. That's where you get to see pictures.
I just wondered.
Yeah, I mean, actual pictures of things mapping to the genome. But here we're really talking more in an abstract sense because we just mashed everything up and stuck it in a test tube, and we're trying to piece it back together. And you do start by mapping it to the reads to the genome. But then when you start building up this information, your alignment file, which typically with a lot of the mapping programs come in the form of a SAM file or a binary version of that, a BAM file, then you have a whole bunch of information about the reads, where each one hit on the genome. And the reality is you'll have a lot of overlap. You'll have a whole bunch in certain areas of commonly highly expressed genes, and other areas that just get a few. And then a few areas where you just get like one hit or two hits and just out nowhere. And so, you have to keep in mind that some things are mapped accurately, some things aren't. And to some extent, your cleanup process can determine how accurate your mapping is. And your settings, too, when you run these programs. So you map to the genome, you then assemble the transcripts, and to do that when you have a genome reference is easier in the sense that all the reads are already overlapping on that genome if you can visualize it, but there's a way of gathering that information and putting it all together looking at the junctions and identifying the different transcripts and the genes that they come from. But at the same time, you can make use of annotation information, the information that's already in the best world for you is if you are working with a well-developed system like a Arabidopsis or the human genome, and in which case you have people that have already gone through this process hundreds of times, and you have a lot of information that's been built up on what are the genes, what are the messenger RNAs that are commonly seen, because each gene can produce different transcripts. So all that information can be overlapped with your information and tie them together. But and the programs that we'll be using are fairly sophisticated at tying them together, and it's hard as a new user to understand, and even for me, who've used these a lot, to really know exactly what each step is doing. But you have to get a feel over time of what works, and sometimes you want to err towards gathering information that's new, because you're working with a tissue that most people haven't looked at before, or cell type. And so the transcripts, some of the transcripts could be entirely new. You just don't see them in what's already annotated. But other times, they're the same old stuff that you see in every cell. And so that you can very readily expect that it's going to be the same. But your data, to some extent, will tell you that if you adjust things in a reasonable manner. Mistakes will be made, yes.
Is there some sort of consensus in the industry, if you will, where when you have a genome, it's just a nuclear genome, or will it be the mitochondrial, plastid, nuclear, everything rolled into one?
Well,
or does it just depend?
You can take, again, you can, when you're doing your studies, you can try to select for different signals, but generally it's hard, if you're looking at full-length messenger RNA, to some extent it's all going to be in there. But as far as your reference goes, most of the time you're going to be looking at the main genomic nuclear genome genes. That's what you're going to be mapping to, and so that's the only thing that you'll really see. Sometimes, though, you may include the mitochondrial plastid for Arabidopsis, and that's often a good idea too, because sometimes those genes will look like some of your nuclear genes, and you don't want to, if you don't need to, misidentify them. So, one of the things that you do, as I said, you can once you have your transcripts, they can be linked up with the annotation from the genome, or that is tied to the genome also. But as I also started out in the beginning to say, in some cases, if you don't have the genome sequence to work with, and some of you may be in that position, in whatever system, if you're working with a copper type of butterfly, one of the little ones, there seem to be a lot of different varieties. Is it sequence? I don't know, but maybe not. Maybe there's no solid reference for what you're looking at. A lot of new species floating around here in southern Arizona. So new in the sense at least that they haven't been sequenced before. So, and Eric took advantage of that, looking at the creosote bush. But, And that can be very interesting, but it's a slightly different challenge. You wouldn't be using tuxedo to start out with, unless you're doing genome sequencing at the same time and assembling and annotating that genome in a very reliable manner. So, you know, these are all things to keep in mind. I can't spend too much time on them, but your ultimate goal is not only to identify what your RNA is in your sample, but also usually you're looking at differential expression. You're comparing one tissue type to another tissue type, or one time point versus another time point. One cell type even versus another cell type. In my studies, I was looking at nuclear versus cytoplasmic RNA. So there are a lot of different things that you can be looking at. Ultimately, though, the difference is often what you're interested in. And that's the focus of RNA-seq studies in many ways. And then in the end, what does it all mean? Well, that you're supposed to be biologists. I think you're biologists. I don't really know you guys, so. But that's the biologist's job. Then your biologist side needs to kick in full time and start looking at, well, I have a list of genes that are expressed differently here. What do I do with that information? And then there are tools for analyzing the gene ontology. What are all the genes, and what do they represent as types of genes? What types of genes are being turned on and turned off? Which types of genes are being turned on especially in this, that is affected much more than what you, than just if all genes were equally upregulated or downregulated, all types of genes. So in some of my studies, I often found that stress-related genes were highly represented in the RNA samples. That what I was seeing, but also specifically being different between nucleus and cytoplasm. But and that was an interesting finding for me. This is the kind of information that eventually you can work towards. But today we're working more with the nuts and bolts. And the first nut and bolt process here is just an example of read cleanup. As I said, we're not doing that today, but it's important to note that you can analyze your data. And one of the things you can also do with your reads is you can just look at them. If they're in the data store, you can just click on a file, and any file, and it will open up and you can read what's in that file. Sometimes you may have no idea what the hell you're looking at, but other times, I'll give you an example at some point here, you'll actually see the sequences. And if you get used to looking at your data, you'll start to understand, well, you know, whether things are going right or wrong.